Hello my friends, this is Dr. Kazmi. Welcome to my YouTube channel. And as I in one of my vlogs earlier, I talked about brief introduction to the three different types of vaccines which have recently become available in the market, and these vaccines are related to the COVID-19. In particularly in today's vlog, I'm going to talk about the University of Oxford and AstraZeneca vaccine, which is making ripples in the news because this is one of the first vaccines which can be stored at normal refrigerated temperatures, that is between 2 to 8 degrees centigrade. And that is actually a ray of hope for the developing countries where the infrastructures for keeping the vaccines at sub-zero temperatures is not widely present. So there was a fear about whether these vaccines would have any practical importance in the developing countries or not. So recently, when the University of Oxford and AstraZeneca released their trial results, that has come as a very encouraging news. Now, let me tell you that as far as the AstraZeneca and University of Oxford vaccine is concerned, this is a different type of vaccine as compared to the Moderna vaccine or the Pfizer BioNTech vaccine, because in contrast to the messenger RNA vaccine, this is actually a DNA vaccine. So what they have done in this particular vaccine is that they have taken the adenovirus, and what they have done is that inside the adenovirus, which is a DNA virus, they have taken the viral DNA out and they have made an artificial DNA which is actually made by the reverse transcription of an actual messenger RNA of the COVID-19 virus. So the COVID-19 virus, when it goes inside the human cells, it is incorporated into the human DNA by a process which is known as reverse transcription. So the same engineering technique has been used, and the viral messenger RNA has been converted into DNA, and this DNA, which is actually an artificial DNA, it's not the real DNA. This has been incorporated inside the adenovirus, and what would happen in this vaccine is when they would inject the person with this vaccine, they would be actually injecting the adenoviruses, which these adenoviruses would be having the fake DNA, and this fake DNA is actually the DNA which has been translated back from the messenger RNA of the COVID. So when it will go inside the human cells, it will go inside the DNA, and then it would order the human DNA to make messenger RNA, and this messenger RNA, by the help of the ribosomes inside the cytoplasm of the cell, would make the spike proteins. Only the spike protein, not the rest of the virus machinery of COVID-19. Why? Just the spike proteins. And when these spike proteins would be released, they would be highly antigenic. They would invoke an antibody response. A lot of antibodies would be produced, and whenever then that person, if that he gets infected with the real COVID-19 virus, this army of antibodies, which would already be there, would attack the virus, neutralize it, and destroy it. So as far as the initial results of the AstraZeneca Oxford University virus vaccine this COVID-19 vaccine is concerned, they conducted two trials. One was in Brazil and the other was in UK. So in total, they had 22,690 volunteers. The trial in the UK had 12,390 volunteers, and the one in Brazil had 10,300 volunteers. So AstraZeneca then pooled the data from both trials so they could have sufficient numbers to compute the efficacy in a statistically meaningful way. So in the combined trials, in the combined results of these both trials, they ended up in probably 100 and if I'm not wrong, 131 COVID-19 cases in their interim analysis, which they think was sufficient for reporting interim results on the vaccine efficacy. So interestingly, what they had done, they had split their trial in equivalent of 4 trial arms. So in the Oxford University AstraZeneca vaccine trial, they split their trial into equivalent of 4 trial arms, and that part of that was due to experimental error as well. The first cohort in Brazil received the equivalent of 2 full doses, which was given roughly 1 month apart. the second cohort in Brazil acted as the control for the first cohort, and that received a meningococcal vaccine plus saline placebo. Now, the third cohort, which contained 2,741 cases in UK, they received a half dose in the initial vaccine, and that was followed by a full dose 1 month later. While the fourth cohort acted as a control for this third cohort and received the meningococcal vaccine only. Now, then the question is, how do you compute the efficacy of vaccine? What they did was they computed the efficacy of vaccine by counting the number of volunteers that got COVID-19 and seeing who got the trial vaccine and who got the placebo. Now, the efficacy of the two full doses, that was the regimen A, was calculated by dividing infectees from the second cohort by the total number of infectees from the first and the second cohort. While in UK, the efficacy for the half dose plus full dose regimen, which was the regimen B, that was calculated by dividing infectees from the fourth cohort by the total number of infectees from the third and the fourth cohort. And on the basis of this, then they calculated the overall efficacy by taking infectees from the cohort 2 and 4 and dividing them from infectees from all the cohort. Now, what they found out that the half dose plus full dose regimen, which was given here in UK, that was a result of a mistake. But this ended up in a huge role in the outcome. We will discuss that later. Now, the first bit of the good news in this particular thing is that no hospitalization or severe cases of the disease were reported among volunteers who received the vaccine. So the data says there were a total of 131 COVID-19 cases in the interim analysis, and the overall efficacy of both regimens was 70%, because it was found out that it was 62% efficacy in Brazil, 90% in UK. So they calculated the overall, which came out to be 70%. So if the interim results hold, then the half dose plus full dose regimen, which was used here in UK and where the efficacy was 90%, then it simply means that the best option would be to give a half dose first vaccine followed by a full dose, and that is quite closer to that of the Moderna and Pfizer as more data continues to be accumulated, because Moderna reported a vaccine efficacy of 94.5%. Pfizer in their final report reported a efficacy of 95%. So the British arm of the AstraZeneca Oxford University has reported it to be 90%. So they're very close to one another. So that seems like to be a happy accident, because by trial the first vaccine was given at half dose, but the efficacy, overall efficacy, was more as compared to those who got the full strength first dose plus full strength second dose in Brazil. So I think for now we are going to give AstraZeneca the benefit of doubt. Most probably this half dose has turned out to be a happy news. But I would quote President Ronald Reagan, the former president of the United States. He said, "Trust but do verify." So I think why should we care about the AstraZeneca vaccine when we know that we have already got two other vaccine candidates that have performed remarkably well with an overall efficacy of 94.5% and 95% respectively. So what I think from a purely pragmatic point of view, none of these vaccine manufacturers, whether it's Pfizer or Moderna, they can manufacture sufficient doses for the entire world. I think the entire world population stands at 7.5 billion, not even by a long shot. So even if they could, let's say, produce all these vaccines for 7 billion people, you wouldn't necessarily want to pull all your eggs in one basket, would you? So a single point of failure is what brought down the Death Star. So the reality is that we are going to need several vaccine manufacturers to be producing their vaccine at maximum capacity to meet the worldwide demand. Second, this new vaccine only requires conventional refrigeration. So I think the University of Oxford and AstraZeneca has reported that their vaccine requires 5 degrees Celsius, which is the normal refrigerated temperature for retaining stability during transportation. And this is a very significant advantage compared to Pfizer, where they require ultra cold temperature requirements of minus 70 degrees centigrade, and Moderna needs up to minus 20 degrees Celsius. So this would be quite difficult, particularly in low-income countries where access to basic refrigeration may be a problem. So Well, I think the most important point is that everyone is pretty much targeting the same protein in the virus, the spike protein. So right now, the different efforts they differ in terms of the vaccine delivery and vaccine production platform, but everybody agrees on the basic viral target. So now that we have got data from three major clinical trials, one from the Pfizer, one from the Moderna, one from the AstraZeneca Oxford. So it appears that these vaccines are offering a definite protective effect that is at least 90% in the short term. And this is amazing news because up till 6 months ago, we didn't know if that would be the case. I mean, and considering everything else surrounding this virus, I mean, I would say let's count our blessings on this one. It didn't have to be this way. And okay, I hear some of you would be saying that we still don't know anything about the long-term efficacy of the vaccine or the long-term side effects. Well, we can't defy the time-space continuum. I'm sorry to inform you, but the virus is new to us. We all know, and we have been only studying it for how many? 10 months now. So the fact that various vaccine developers were able to take the viral genome information and then used it to develop these trial vaccines for this novel coronavirus in such an abbreviated time scale is it's nothing short of a miracle, I would say. And the fact that we are starting to see phase 3 clinical trials data and they have been uniformly good, I think it has to be celebrated. Now, what we can't do anything about long-term efficacy data for the time because in order to know the long-term efficacy, we need to follow up these volunteers for a longer period of time. And you know how consumer products can be stress tested to see how long they can be expected to last. I mean, we can develop a robot that can simulate sitting down on a chair probably 1 billion times, but we can't do that with the vaccines. The reason we could get more rapid primary endpoint data on vaccine efficacy in the coming phase 3 trials with that these trials have been unprecedented in their scale. The number of enlisted volunteers is simply massive. I mean, see all 30,000, 12,000, 10,000. That's quite a massive figure. So then getting sufficient numbers of infected volunteers to compute basic vaccine efficacy, I think it could be reached sooner as well. And frankly, because there is so much community transmission where these trials took place, like in Brazil, we got the required number of infectees much sooner than we thought. So, but again, I would say this does not address long-term efficacy. So we are just going to have to stop fetishizing that. It's not that important for the timing. It's just that we can't do anything about it for the timing except to follow up these volunteers for a longer period of time. Similarly, then there is a whole question as to whether the vaccine truly keeps people from becoming infected and infectious. Now, these trials, they didn't assess all active viral infections, only the one from the people who were showing visible symptoms. So do these vaccines also stop asymptomatic transmissions? Well, we don't know. We are not 100% sure. Anyhow, now if people around the world were being smart and we had controlled this virus to the point that where we were only dealing with minor outbreaks here and there, then we could have the option of waiting for long-term efficacy data and other important parameters before rolling out these vaccines. But instead, in the current dumpster fire around the world, we have a record number of people becoming infected and dying each and every day, and the capacity of the medical care system around the world is at a breaking point. So, because we are a long way from reaching herd immunity levels, this has for no foreseeable end in sight. I don't think so. And case fatality rates will start to creep up again once hospitals are at their maximum capacity and our medical personnel can no longer keep up with the demand. So this is not the time to act with timidity and to let vaccine hesitancy rule your life. Life does not return to anywhere near normal until enough people roll up their sleeves and get these vaccines. So I would thank God, at least in the short-term efficacy, the vaccine so far looks impressive, and let's hope that the long-term efficacy remains good. and the rate of adverse effects remain as low as what we have seen so far. So thanks for listening to me. This was a short introduction to the University of Oxford-AstraZeneca vaccine and its trial, and what does it mean for the overall picture in the coming days. So hopefully, as these vaccines, they hit the market, they are on the shelf, so the governments would be subsidizing to buy these and vaccinate their populations, and hopefully in the coming year we would see a drop in the number of cases, and I hope that there would be no third wave. Thank you for listening to me. If you are visiting my channel for the first time, do subscribe, like the videos, and if you like it, do share it with your friends as well, and do press the bell icon so whenever a new video is uploaded, you get the notification. Have a good day. Stay healthy, stay safe. Bye bye.